with viruses, viruses are not considered cells. So we don't consider them prokaryotic or eukaryotic cells. They are off in their own category. So with viruses, really all they are is some genetic material, either DNA or RNA, surrounded in a protective covering. All viruses will typically have their genetic material surrounded by a protein covering called a capsid, and then some viruses will have an additional outer covering called an envelope. But they really are just genetic material surrounded in a protective covering. They are not independent, meaning they depend on living cells in order to replicate themselves. So one virus will always just be one virus unless it has the ability to infect a cell. So they are dependent on living cells. Once they infect a living cell, they will essentially hijack that living cell and manipulate that living cell in order to produce more viruses. They don't themselves have the metabolic machinery in order to produce more of themselves. So that's why they have to infect a living cell, hijack that cell's machinery, and then manipulate that cell into producing millions of more viruses that will then go on and infect future cells. So viruses are not considered cells. However, they are essentially particles that are able to infect and manipulate living cells. So when we talk about microorganisms, we typically refer to them using their species names. So when I say things like Escherichia coli or Bacillus anthracis or Staphylococcus aureus, those are all species names. And with a species name, it is actually constructed using a two-name system. So within the species name, which is a two-name system, the first word is going to be the genus, and then the second word is going to be the epithet. And when you say the genus and epithet together, that's where you get the species name. So when we say Escherichia coli, Escherichia is the genus, coli is the epithet, but when we say it all together, that's where we get the species. So anytime you're working with species names, you want to make sure that you properly construct them. So anytime you're typing up a species name, you always want to make sure you italicize it. So an entire species name should always be italicized. If you were handwriting it to show that it's italicized, you would need to underline it. Also, capitalization is important. The only letter that should ever be capitalized is the very first letter in the genus. So if we look here at the species, Escherichia coli, we have the genus name, which is Escherichia, and the epithet, which is coli. It's all italicized, as it should be, and only the very first letter of the genus is uppercase. Everything else is lowercase. We have the option to abbreviate these species names, as some of them can obviously be quite long. How you abbreviate it is you always take the first letter of the genus, it still needs to be capitalized, and put a period and then the entire epithet. So the species Escherichia coli then becomes E. coli, and Staphylococcus saprophyticus becomes S. saprophyticus. So please make sure you know how to break down the species names as far as genus and epithet. Remember to always italicize the species name and always have that first letter of the genus capitalized, whereas everything else is lowercase. Species are relatively broad categories, and we can further subdivide a species into different components, which we call a strain. So a strain is a subgroup of a species with one or more characteristics that distinguish it from other subgroups of the same species. So when I say E. coli or Escherichia coli, that is a species, but there are hundreds of strains within the E. coli species. All these strains have similarities, and that's why they're obviously in the same species. However, they typically have some unique characteristic that necessitates them being put into their own separate strain. And that could be that they're resistant to a specific antibiotic, or they maybe require a particular nutrient or have a certain type of receptor. So for example, Escherichia coli is a species, but there are different strains of E. coli. These two strains, E. coli strain K12 and E. coli strain 0157H7, have a lot of similarities. That's why they're in the same species, being E. coli. They're both rod-shaped. They both prefer to live at 37 degrees Celsius. They both can survive in the presence and absence of oxygen. But they have one very important difference, which necessitates them being put into different strains. E. coli strain K12 actually produces vitamin K for us, which is beneficial and helps with blood clotting. E. coli strain 0157H7 actually produces a toxin that leads to profuse vomiting and diarrhea. So while these two strains are very similar and belong in the same species, the fact that they produce different chemicals necessitates them being placed in different strains. So a lot of the terminology that we use in microbiology actually dictates what the microorganisms are going to look like under the microscope. So a lot of bacteria are named based on what they look like under the microscope. Not all. For example, Escherichia coli is not named for its appearance under the microscope, but things like Streptococcus pyogenes and Staphylococcus aureus are. So sometimes you can kind of predict what a bacteria is going to look like under the microscope before you visually see it. So a very common genus within microbiology is the genus Staphylococcus. So we need to kind of break down what Staphylococcus means. So in micro, coccus means circular. So anytime you see coccus, you'll just think bacteria that are circular in shape. And staphylo means clustered. So anytime you hear Staphylococcus, you can imagine that these bacteria would essentially be clusters of circles under a microscope, which is what we're seeing here in this image. This image is a picture of Staphylococcus epidermidis. That's a species name. But the genus Staphylococcus tells us that these bacteria are going to be clusters of round circles under the microscope. And it doesn't matter if it's Staphylococcus epidermidis, or Staphylococcus aureus, or Staphylococcus saprophyticus. Any bacteria within the Staphylococcus genus is going to look like clusters of round circles under the microscope. Another very common genus is Streptococcus. So same as the last, coccus still means round circle, except in within the Strepto genus, these bacteria are arranged as chains of round circles. So when we see any bacterium in the Streptococcus genus, we know that under a microscope, they are going to exist as chains of round circles. So this is Streptococcus pyogenes, but it doesn't matter if it's Streptococcus pyogenes or Streptococcus pneumoniae. Any bacteria, again, in the Streptococcus genus is going to exist as chains of round circles. A third very common genus is the Bacillus genus. Bacteria that belong to this genus are things like Bacillus anthracis that causes anthrax, Bacillus subtilis, Bacillus megatherium. These are all very common species of bacteria. What they all have in common, though, are that they have a rod-shaped appearance. So in micro, Bacillus means that the bacteria have a rod-shaped appearance, which is obviously different from coccus. Coccus means that the bacteria have a circular shape. Bacillus, again, means that the bacteria have a rod-shaped, and we can see these little rods here. Again, this is Bacillus anthracis, but it doesn't matter what species we're talking about. Any bacteria in the Bacillus genus is going to have this rod-shaped appearance. Another genus is the Vibrio genus. So bacteria in the Vibrio genus are all going to have a curved rod appearance. I always remember the curved rods as they'll look like little V's. So this specific bacteria is Vibrio cholera, which causes a very severe GI infection. But this bacteria is within the Vibrio genus. So even before seeing it under the microscope, we know that it's going to have a curved rod appearance under the microscope because of the genus that it exists in. So any bacteria in the Vibrio genus will exist as a curved rod under the microscope. And then the last one is Spirillum. Within the Spirillum genus, these bacteria are going to have a spiral-shaped appearance. So this is Spirillum minus, which causes a disease known as rat bite fever that happens in Asia. But this bacterium exists as a spiral. So again, it doesn't matter what species we're talking about. Any bacterium within the Spirillum genus is going to have this spiral shape. So definitely want to make sure you understand terminology and what those terms mean in kind of layman's terms. So again, coccus is going to mean circular. Bacillus is going to mean rod. Vibrio is going to mean curved rod. Spirillum is going to be more of a loose spiral shape. And then there's also different arrangements. Staphylo means that there's a clustered arrangement, whereas something like strepto would mean that it exists as a chain. So the next points that we'll talk about have to do with the history of microbiology. So traditionally, in the when microbiology kind of first came about, most individuals believed in this theory called spontaneous generation, which is going to sound unbelievable, but at one point people did believe this. In spontaneous generation, it was the belief that invisible life forces present in matter led to the creation of life. So essentially, spontaneous generation was this belief that something non-living, like a pencil or a desk, whatever, could release a magical force and produce a living organism, like a cat or a dog or a bacterial cell, whatever. We obviously know that's not true, but during these times, people believed in spontaneous generation, and they really believed in what we kind of refer to as abiogenesis, that something non-living could produce a living organism. We obviously know that's not true nowadays. Nowadays, through modern day science, we typically abide by the theory of biogenesis, that living things produce other living things of the same kind. Bacteria produce bacteria, humans produce humans, dogs produce dogs, etc. So spontaneous generation and abiogenesis is out. Those theories have been disproved through science, and what we currently abide by is biogenesis. Another thing that we currently abide by is the germ theory of disease. This is the theory that microorganisms invade other organisms like humans and cause disease. So the COVID-19 virus invades a human host and causes the disease that we know as COVID. During times of spontaneous generation, if someone got sick, it wasn't because they were infected with a bacteria or virus. It was because of some force within their body that was making them sick, maybe from karma or some magic force, whatever it be. We obviously know that that's not true as far as infectious diseases. If you have tuberculosis, it's because Mycobacterium tuberculosis has gotten into the lungs and you have now had that condition. It's nothing to do with any type of magical force.